Good morning, and thank you for the introduction. But thank you also to Nigel and the organizers for inviting me to talk today. So as was described, I'm going to describe my group's research in trying to develop a pipeline so that we can link specifically proteomics and structural biology. But as you'll notice from the very top of the slide, my research group actually is residing inside of EMSL, which is a sister user facility to JGI. And for those of you who may not already be users and don't know very much, I wanted to spend a couple minutes to just kind of show a little bit of the breadth of the type of research that we can do that's very complementary. And it also kind of gives a background to the structural biology capabilities that I'm going to get to in a few minutes. So at EMSL, there we go. EMSL is a national user facility from DOE BER, but we wind up focusing on really molecular characterization using an array of multimodal techniques. So you can see here that we have a wide array of different capabilities. We are very well known for proteomics and metabolomics, mass spec and NMR capabilities, but we also have microfluidics, we have high-performance computing, optical imaging, both in terms of label-based and label-free methods, as well as electron and ion imaging. And in terms of gaining access to the user facility, there's 4 different mechanisms that we have. The FICUS proposal you've seen highlighted on the screens the last couple days. That one, the letter of intent, is due next week, and the full proposal is at the beginning of May. But we have 3 other tiers of science theme, general, and rapid. We've missed the cutoff for the science theme ones, but that's an annual call, so it'll open up again next year. But the rapid and general provide 2 mechanisms where you can apply or submit a proposal at any time during the year, and you can be considered for access and awarded time. The rapid proposal is a great one for getting introduced to the lab, especially since you can use it for things like getting preliminary results for a proposal, or if you get review comments back from a manuscript where a reviewer is saying you need to do one more experiment and you don't have that capability or expertise, that's kind of the idea behind the rapid proposal. And then it gets you used to the facility and gives you a way to go into the other ones. Other access mechanisms. But as I mentioned, we have a wide array of capabilities in a range of different areas. And so in terms of just zooming in on kind of one of these capability areas, these are just a subset of the instruments that we have for the bioimaging and structural biology capability, where we have many electron microscopes, as well in order to be able to do reconstructions from individual particles to get atomic structure, as well as whole cells. And we also have top-down and native proteomics to look at post-translational modifications on proteins, oligomeric and assembly states, NMR and EPR for other de novo structure determination, molecular modeling, whole bunch of fun tools, all at the molecular scale and at the size of the proteins and whole cells. We can also work with larger communities. And with helium ion microscopy, we can start to look at the topology of 3D architectures, as well as with FibSem, we can look into depth architecture of overall communities as well. So there's a lot of great capabilities that we have. One of the newest additions to it is microelectron diffraction. And this is a technique that allows us to get atomic resolution structural details from 3D crystals, either protein or peptides, that are conventionally too small for most X-ray research, but too big for most typical electron microscopes. And so using microelectron diffraction, there's a number of other benefits to it. This was the first example that we had of the utilization of our instruments. This was in collaboration with a group from UCLA, Jose Rodriguez, and we wound up getting 1.1 angstrom resolution off of our microscope from these crystals, and he wound up optimizing that and ultimately getting to 0.76 angstroms, and this was just released a couple months ago in Nature Structural and Molecular Biology. So that, all of those capabilities that I already just described, including the microED, are completely available to users right now, and you can apply through it through the FICUS or general and rapid. But going to the original point of the talk of trying to create a pipeline to link proteomics and structural or functional analysis, this is really trying to build off of the strengths of EMSL. I mentioned that EMSL has a long history in metabolomics and proteomics. So we work with a particular organism that one of my lab members we got a user proposal to be able to get access to the mass spec and NMR. We wound up doing runs with the experts in those areas at EMSL and profiled over 287 lipids, some showing upwards of 100-fold change under certain conditions. Proteomics profiling, we did both global and phosphoproteomics. And with the ability of getting a map like this, of looking at the overall abundance and changes in proteins, this is kind of where we started to realize there was a bottleneck in linking proteomics and structural biology. Here you could wind up seeing a number of proteins that you would expect to see changes in given the conditions that we used for these experiments. But then you wind up also in green and orange, being able to start to see other things that come into play that were kind of not expected or completely unknown. Everything in orange is actually a protein that has unknown structure function. In fact, of the top 10 upregulated and top 10 downregulated, you actually have 8 of those proteins are completely unannotated. In addition, there's other proteins of interest. You see in blue the glutamine synthetase. This is an interesting protein because this organism has only a single isoform of glutamine synthetase, and it's a type 3 isoform, which is very rare to find in nature that you only have a type 3. But this organism is very interesting. If you wind up incubating the cells with known inhibitors for glutamine synthase, this organism can take on about 1,000 times higher concentration than the concentrations that are known to kill all other algae and plants, and this organism just slows down growth. The known inhibitor of glutamine synthase, and this organism has only a single copy of glutamine synthase, and it's pretty resistant to it. So biochemically, it's very interesting. Structurally, it's also interesting because you'll note that across the top right, you can see all the different known structures for the 3 different classes of glutamine synthase. And the prokaryotic forms are all known, but the eukaryotic type 3 isoform is completely unknown. There's no known structural knowledge for it. Interestingly, the 3 different isoforms of glutamine synthase wind up having different oligomeric states, 2 stacked rings of hexamers or 2 stacked rings of pentamers. And between class 1 and 3, which both have double stacked rings of hexamers in these structures, you actually have an opposite orientation of how those come together. Instead of head-to-head interactions, you have tail-to-tail. So you have different assembly. There's a lot of interesting questions with the type 3 isoforms. And the version that we have from that organism I was talking about is eukaryotic type 3. It has limited homology to the prokaryotic one, so we can run homology models in order to give rise to some hypotheses that we might be interested in. And you'll note that in the square box, you can see a region that if the homology model is true, we actually have a deletion of amino acids. And those alpha helices that extend up there are described in publications as stabilizing the hexameric structure. In addition, we have in the oval that's circled a region where we have additional densities or amino acids that are added. This is nearby the active site region. So one or both of these, the deletion or the addition in these areas, could potentially play a role in affecting the activity of it and giving rise to the additional resistance of it, if this homology model is correct. So what we wanted to do is, after getting the proteomics data, identifying that this and many other interesting proteins are targets of interest, we then wanted to develop a pipeline to be able to link this identification of targets to overall protein and structural, to functional and structural assays. So this comes back to the overall idea that my research group was interested in this, but we also noticed that users who were coming to us to do proteomics data sets would identify proteins, and if they came up to me asking, we've identified a protein of interest we want to do structural work on, I would have to tell them at the time, I'm sorry, I would love to work with you, but we don't have the capabilities to do that on site. We need you to go back to your own lab, develop the expertise if you don't already have it in genetics, molecular biology, biochemistry, to take that target, get a gene, express it somehow, and then purify it. And once you have a protein of high enough yield and purity that's suitable for structural approaches, then I'd be happy to take it and work with you. This was a huge bottleneck that we noticed. And so this really drove the need for developing a pipeline to link these things. And so what we wound up focusing on was creating a cell-free pipeline. And we have 4 different stages to that pipeline that allow us to optimize for solubility, for expression, for purification, scale up for the different approaches. And the very first step actually starts with 25 microliter reactions. So we can get the gene of interest, do a very quick double-stage, 2-stage PCR process to create a PCR template that we send in, do screening with it, and on these very small scales, we can screen for whether or not the gene actually expresses, and 2, whether or not it's in a soluble form that we could potentially do purification with it later on. There's obviously some proteins that are very difficult to pull out, so we use a nested PCR approach in order to pull out these hard-to-amplify genes in order to get our overall PCR products. But once we wind up knowing that at the small scale, the mini scale that we describe, that we can see expression, then we start to scale up. And the first step with that is to generate a stable plasmid with the gene of interest in a fully optimized form. We use a modified form of a PEU vector, and we wind up getting the samples in, the genes into the constructs by a number of different methods. Here's showing Gibson assembly. But once we have that, then we can actually do expression and some initial purification steps, which you can see in panel C on the far right. And this is at the MIDI scale. So we've scaled up from 25 microliters up to about 200 microliters. Still very little reagent, so we're not costing a whole lot, but are able to do a wide array of different tests for purification. Now from there, once we know that we can purify it, we can scale it up. And here's a description of the overall workflow for this pipeline now. And you can see we can start with a target, get it into a plasmid, do in vitro transcription in a test tube. In the same test tube, do in vitro translation, then go through purification. You can see a very clean purification. This is actually the glutamine synthetase protein that I was talking about earlier. So that protein that we identified from the proteomics, we sent into this cell-free expression pipeline. Once we get a purified protein, we can send it in and do functional assays on it, look at the enzyme activity, show that it's fully functional. And then in this case, when we scale up to the maxi scale, we have enough protein yield that we can send it for cryo-EM and other functional and structural approaches. But you can see a cryo-EM image at the bottom, the reconstructions that come out of that, and then the 3.5 angstrom resolution atomic model that we get from that. And when we scale up to the mega scale, now we have enough protein in order to be able to do X-ray crystallization. This platform is nicely versatile. We've been able to demonstrate expression from plants, fungi, yeast, bacteria, and even some mammalian cells. So very nice that way, but the real power from it is actually being able to utilize this in order to address some science questions. If you remember back to that homology model, the model that was created based off only 30% homology to the prokaryotic version, the eukaryotic one, if that homology model was correct, would be a double-stacked ring of hexamers. In this case, the cryo-EM clearly shows a single reconstruction of a single layer, so it's a hexamer still, but it's only one ring. And if you look at the X-ray crystal structure, the unit cell that is present is too small to physically fit 2 copies of it. So you have 2 different structural approaches confirming that this molecule does not stack in a double-stacked ring format. And in addition, we've done native page assays incubating nucleating with different ligands and so forth, also structural approaches on those same ones. None of them show any higher order oligomerization in assembly states. So this is really intriguing, why this particular version of isoform 3 of glutamine synthase exists only as a hexamer, and all other known structures exist as double-stacked rings or not, is giving rise to additional research. But this isn't a one-hit wonder show. Everything that you see here in gray, and this slide is about a month old, hasn't been updated. Everything that's not gray, it has already been expressed at some level and is either in further optimization for purification or in direct structural approaches. You can see there's a lot of ones already in blue. A couple of those are the CCMK, which is a carbon dioxide concentrating mechanism protein that's from a bacterial source. For those of you in the front rows, you may be able to see there's an array of little hexagons that are seen in the left picture. This is a 2D crystal where this protein, we've been able to basically assemble it into a single layer, but in that layer you have proteins stacked side by side like a normal crystal. Able to do 2D electron crystallography on it. You can see the unit cell in white next to it, and then the overlay of the atomic model that in red. And then PDX, a protein involved with vitamin B6 biosynthesis from plants, able to do single particle analysis on it to get a structure as well. So this pipeline is really powerful for being able to produce the protein in the quantities and purity levels that are needed for these structural approaches. But I don't want to give the impression that we're only using it in order to be able to do structural work. In fact, one of the proteins that we wound up working with, an ice-nucleating protein, these are proteins that are naturally found on the outer surface of bacteria, and they help nucleate ice at very high sub-zero temperatures and cause frost damage to crops, as well as play a role in aerosol formation, as well as cloud and snow formation up in the upper atmosphere. These proteins are difficult to work with. They're large. They have a membrane association fragment on its N-terminus. They tend to aggregate very easily. So it poses some issues with structural approaches. We're working on that area, but I didn't want to focus on that area. What we've done is cell-free expression with these. We've been able to express them, get them soluble form, able to work with them. And then we wound up making multiple different constructs of them. We've made a version that has the full-length sequence as well as one that's missing the N-terminal sequence, and we wanted to see functionally, are these proteins, one, that are cell-free expressed, able to nucleate ice, and two, if they are, did they have any change when they're missing the N-terminal membrane-bound portion? So in this experiment, we wound up using one of the microscopes at EMSL, an environmental SEM. This is a scanning electron microscope that you can operate not under high vacuum, but you can have it at about 20 torr, so you can actually introduce water vapor. In these experiments, we wound up taking a sample of buffer or buffer with the ice-nucleating protein in it, depositing that on a substrate, drying it down, putting it into the ESEM, and then cooling that dried substrate down, and then introducing water vapor in order to raise the relative humidity. And that way we could control the amount of water that was there and see if we could identify the point at which ice nucleation occurs. And what you'll see here is that starting on the left with the buffer or the control, you wind up having some artifacts or debris that are there, but even that debris doesn't allow the ice to nucleate. Everywhere else, as you increase the relative humidity, bring in water vapor, all you form are these water droplets from condensation, and then at the water saturation point, everything is fully saturated with these condensates. However, if you add buffer with NZ in it, now you actually nucleate ice at low temperatures. In this case, you can see ice nucleating already in the second image, and then it just grows and grows. So the addition of the NZ is, that was self-free expressed is fully functional for forming these ice nucleation events. And here you can do this with a range of different temperatures, different relative humidities, identify at which point you wind up starting to see nucleation. You can create this plot where effectively the lower the value, the better at ice nucleating function you can see in these. And you can clearly see, you see the diagonal line. That's the natural line for water vapor at low temperatures between natural ice formation and liquid form. And so you can see that buffers containing liposomes or just regular are at that line or above it. Anything with the ice nucleating proteins is actually below. So it's nucleating ice before you would naturally have it. So I've kind of taken you on a journey of being able to show that we can now link these proteomics data sets and start to do some structural and functional analysis on it. But I don't want this to be viewed as just something that we're intending to roll into the EMSL user program, and it's only going to have impact there. What I'd really like to see with this is that it gets used as a vehicle to start to integrate and leverage all the strengths of the multiple different user facilities that you have available to you. So one way to do that is, as I mentioned, we at PNNL and EMSL have to do manual gene synthesis as well as get them into the plasmids and so forth. That's the major strength of JGI, in addition to doing sequencing, is the DNA synthesis. So you have a direct relationship there that we could do proteomics, identify targets at EMSL. The users or us could send those targets to JGI to do DNA synthesis. We then do cell-free expression on those and then send those expressed proteins into structural pipelines at EMSL, at synchrotron light sources, other places, functional annotation, wherever, and it really creates a resource that's centralized in order to link these things, but builds on the strengths of all the disparate institutions, as well as computational biology. You can use these targets, do database mining and so forth, identify optimized clones from sequences, and then use those in your cell-free expression to create the isozyme of interest that has more functionality than the original wild type. So with that, I'd like to wrap it up and take any questions. You can see all the people who played a role in this project, and I'd also like to thank the DOE for funding.